Hello, everybody. My name is Omai Garner. I am a professor and section chief of clinical microbiology. I am the director of point-of-care testing in the Department of Pathology and Lab Medicine at UCLA Health. And my talk today is something I'm very passionate about. It is the lab's role in advancing equity. So we have three learning objectives for today. One is to really understand how lab testing can contribute to healthcare inequities, then be able to describe how equitable access to lab testing can positively impact patient care, and detail the numerous ways that laboratories can be involved in lab equity. I want to start with a slide that I have really taken from Dr. Carissa Culbreth, who is leading in this field, on this idea of defining health equity and health disparities. So this is a slide that really outlines health imbalance. Health equity is this idea that every person has the opportunity to attain their full potential of health, and no one is disadvantaged from attaining this potential due to race, ethnicity, age, disability, gender identity, sexual orientation, nationality, socioeconomic status, or geographical background. We balance this with health disparities, and health disparities are preventable differences in the burden of disease, injury, violence, or in opportunities to achieve optimal health experienced by socially disadvantaged racial, ethnic, and other population groups and communities. So how does this connect to lab testing, right? I think a lot of people think about health disparities in things like access to care, right? If you live in a community that doesn't have good access to care, hospitals, urgent care clinics that are available, then by definition, you will be experiencing health disparities. But to highlight how the lab plays a role in this, I really want to talk about the global access gap to lab testing. So 47% of the global population has little to no access to diagnostics. 1.1 million premature deaths in low-income, middle-income countries could be avoided annually by reducing this diagnostic gap for 6 priority conditions. And I'm going to show you some data around this. This is a publication from The Lancet. So these conditions are diabetes, hypertension, HIV, and tuberculosis in the overall population, and hepatitis B and syphilis for pregnant women. If you're able to increase access to diagnostics, this has a very favorable benefit cost and is favorable for middle and low income countries. So let's really dive into some of this data here. So here we have the conditions on the left: diabetes, congenital hepatitis, congenital syphilis, HIV, and the like. And this first percentage really represents the current diagnostic gap for diagnostic availability for all peoples in that category. So let's take HIV. The current diagnostic cap for HIV diagnostics is 46%, and the current number of annual deaths is around 938,000 deaths for this condition, of which if we had diagnostics at a full coverage, right, at 90% coverage, meaning 90% of people were able to access diagnostics for this condition, the number of estimated annual deaths that could be averted is 250,000. Like, to me, that's staggering, right? When you think about lab diagnostics and access, the important impact that these can have. So if you add all these conditions up, diabetes, hepatitis, HIV, hypertension, syphilis, and tuberculosis, all which have key diagnostics in the space, potentially 1 million lives could be saved. We could avert an annual death of 1 million people per year just by increasing access to diagnostics. This is not just true globally, though. This is also true in the United States. And when we look at this particular publication, we see that average median travel time to a testing site was 20 minutes overall, but that's across the country. But if you actually look at disaggregated groups, whether they were minority groups or uninsured groups, what you saw was a really long increase in that travel time or availability to diagnostic testing. And any increase in travel time to diagnostic testing, or I would argue not even travel time to phlebotomy, but travel time that sample has to take to then go be tested in a diagnostic laboratory, it increases the turnaround time and decreases diagnostic access. So then if you take rural plus insured people, there's a high uninsured, there's a higher incidence of increased travel time, and this, of course, also contributes to decreased access to testing sites in communities of color. So I think we tend to shape sometimes this argument as sort of an outside low and middle income centric argument, country centric argument, but these are disparities that we see in the United States as well connected to lab testing. This especially hit home in COVID. So if you look at the inequities related to COVID, I think the pandemic actually just exacerbated or revealed some of these existing inequities. So in LA County, where my laboratory and hospital system is, if we look at the testing gap, meaning testing availability in Los Angeles County at either comparative majority white communities, communities that had access in LA County, or communities of color, communities that did not have as good diagnostic access. April 30th of the beginning of the pandemic, and we'll all remember, because we all experienced this, the very beginning of the pandemic, it was critically important to be able to figure out who had COVID. There was about a 65% racial or ethnic testing gap in COVID-19 testing availability. Now, move forward another month, and that gap started to close. It moved to about 25% in LA County as our tests grew and the number of our tests grew. But this diagnostic testing gap, this inequitable gap, led downstream to inequities of care and unfortunately increased mortality related to COVID-19. So it's really the pandemic showed how important it is that diagnostic testing availability is equitable, or as equitable as possible. So I think the question then comes up, and this is really what we're trying to address today. Does the clinical lab have a role in addressing health equity and diagnostic access? So ALCS in 2013 put out a position paper that I think highlighted the importance in medical decision-making of clinical lab testing. Clinical laboratory services are the most cost-effective, least invasive sources of objective health information in disease prevention and diagnosis, improving patient outcomes, assuring patient safety, and fulfilling essential public health surveillance functions. So critically important all the way around, and really critical from a health equity argument perspective. And so I like to think on equitable lab testing as a spectrum. And the idea here is that there are a lot of components within the clinical laboratory setting that can assist in equitable lab testing. And this is a team goal, right? There isn't a single person that's going to make lab testing equitable. There isn't a single healthcare institution that's going to make lab testing equitable. This is this idea that if equitable lab testing is an important goal for all of us, then each one of us can work at different points in this particular pipeline to be able to create equitable lab testing overall and improve health and decrease health disparities. So when I look at this spectrum that leads to equitable lab testing, I really try and put it into categories that are relevant for the clinical laboratories. And not just the clinical laboratories. This includes diagnostic manufacturers, this includes researchers, all of us in the clinical laboratory community. So the first part of this is really about novel assay component development, right? If we're going to make a better, cheaper, faster, more available test overall that's going to address some of these inequities, then maybe we need new antigen antibody targeted assays, things that are more sensitive. Maybe we need new DNA and RNA targeted assays, right? Molecular assays that maybe aren't built on the foundation of PCR, that can be expensive, but are built on other sort of nucleic acid amplification foundations. And then we need cheap, accessible readers for that. So there's a group of work that's being done in that component, and I'm going to share some of that work with you today. Then there's clinical test development. This is this idea of patient sample testing, right? Development of validation of clinical assays, taking it from the research into the clinical space. And a lot of us work in this space, right? Partnering with diagnostic manufacturers to create better tests, to create assay optimization. Then there's an entire group of regulatory approval that's necessary, right? Whether this is FDA preclinical and clinical trials or state licensure for different testing. And I know there are groups that are interested in increasing access by increasing the amount of FDA-approved tests that are available. And then finally, an important point to this is clinical test implementation and study. So if a new test comes out, how do we know that it broadens access? How do we know that it can accurately reach out to communities that are disadvantaged? And that requires studying, right? That requires launching new tests, point of care tests potentially, tests that have easier access, and then studying their clinical effectiveness. All of this can then lead to equitable lab testing. So today I want to share a couple of stories with you in the limited time that we have about one novel assay component development that we've been working on, and then some clinical test implementation. So again, I mentioned this is cross-disciplinary, right? This requires lots of people working on it. I'm fortunate enough at UCLA to work with some very talented engineers that are interested in the diagnostic space. So Professor DiCarlo works in fluidics, and Professor Oskan works in imaging and sensing architecture in cell phones, right? Another device that is sort of widespread and can be used for equitable testing. And in working with them, it's interesting in working with engineers as sort of a clinical laboratory person, because they have lots of great ideas, but not necessarily where they should be best used. And so I sort of highlight here a conversation that we had with our engineers years ago. They said, I've got a way to read a 96-well plate on a cell phone. Is that useful? And so my follow-up question was, can it read changes in color? And the engineers emphatically said yes. And so we really started to develop at that point a cell phone-based handheld microplate reader. So there's this idea, and everybody knows working in a clinical laboratory, immunoassays predominate. But we have these very, very large automated machines, or even smaller machines that require high complexity laboratories at time. Or if it's going to be moderate complex, it's going to be a large machine, and that doesn't necessarily lead to good access in testing. And so the idea is, could we make a cell phone reader out of a 96-well plate? And so here, just sort of the basics of how this works. You run your ELISA plate, so there's color within the dish. You load the dish here. There are some LEDs that shine through over the sections, and then there are optical fibers that pick up the signal and then deliver this signal to the cell phone eye of a camera. Now, I like this slide because it just highlights everything good in engineering requires machine learning. And so this is where some of the artificial intelligence works come in to determine which signal is relevant in that space. So here, the reader transmits a signal. It creates a map, right? And you can just imagine these like ODs for any of you who've worked in an immunoassay setting. But then it needs to be calibrated, just like you calibrate an OD, to get the reading to produce a clinical value. That calibration really requires sort of some intricate work in each well on the different levels of positivity in that well. So should you read the inside of the well? Should you read the outside of the well? This is really where the machine learning comes into place to then assist in the diagnosis. And what's powerful about this setting, of course, is that cell phones have computers that can already handle all of this. So we are already living in an age where diagnostic devices like the cell phone potentially have the computing power to be able to be used. So did it work? So we took the handheld reader. I took ELISA plates from my clinical laboratory that had been run on a couple of tests, mumps IgG, measles IgG, HSV1 and 2 IgG. They had been read out already as an OD, reported clinically, and then we compared them overall to what we got on the cell phone as a plate reader, as a value. And what I'll say is that it worked. Right? It worked to the point that it could even be clinically validated as a usable instrument in the space. So it doesn't solve all the problems when we're talking about lab equity, but really starts to get at this idea of cell phone usage for technologies that would be able to add access to care for tests like immunoassays. Immunoassays, again, being the bread and butter of a lot of what we do. We created a graphic user interface. So, we talk a lot about EMR access and electronic medical record access and understanding middleware connections, right? This is a lot of the space that we talk about in the laboratory. And unless you're involved with a large laboratory diagnostic, that also isn't available, right? You don't have sort of an easy way to send signals, let's say, to a clinician to understand what the results would be. This is especially true in the point of care setting. And so we build a graphic user interface that allows you to select the ELISA And then you could imagine a scenario, if you were putting this into clinical implementation, that the result could then sort of be uploaded right away into some sort of My Health or My Chart interface, right? That could then either connect directly to the physician and give those results just like they were coming out of a clinical laboratory. So again, taking advantage of the power of computing that's there to increase lab access. Okay, so we felt good about that, but again, lots of challenges. We're still doing ELISAs and plates, not exactly access forward for everything we want to do. So another conversation, the engineers said, well, that was wonderful, but what else is read with a 96-well plate in the laboratory? So I put my sort of clinical microbiology hat on and asked them if they had ever heard of antimicrobial susceptibility testing. So why is this important? So antimicrobial resistance sort of is a overall pandemic that is happening and is not being well recognized. So every single year, we have more and more deaths across the world attributed to antimicrobial resistance. But the places that have the best diagnostics and the places that have the most drug availability from an antibiotics see way fewer deaths. And so this is highlighted by this map here, that if we look at deaths attributable to antimicrobial resistance every year by 2050, right, projecting out in the future, you can see in North America a substantial number of deaths, 320,000. But in Africa, we're really talking about a log scale higher, right? 4 million deaths attributed to antimicrobial resistance. And some of this will be due to the fact that diagnostic testing for antimicrobial resistance is difficult and is not routinely performed. Thus, it's very difficult then for the treating clinician to know what antibiotic to use. So we wanted to address this from an equitable standpoint in development, in research development as well. So the equity issue is lack of highly trained lab scientists to perform and interpret test results, because the gold standard that we use for this is called broth microdilution. And broth microdilution, 96-well plate, right? Back to something we're going to use a cell phone or something cell phone-like for. So broth microdilution is where you take bacteria and you put it into wells in increasing concentrations of drug. And when the drug is able to kill the bug, then you have what's called a minimum inhibitory concentration. This can be compared to a breakpoint, which offers interpretive criteria for whether or not an antibiotic is resistant or susceptible for a certain bug, right? Staph aureus, Pseudomonas, whatever the bacteria happens to be. So there are automated ways to do this, right? A couple of different companies make automated platforms that are used in our high throughput laboratories, but those are very expensive, prohibitively expensive for a lot of the world. And then to individually read a plate like this, places can set up bacteria and put it into the wells with the drugs, but reading it takes highly trained clinical laboratory scientists. So the question that really came up on the clinical lab side is, we know the reader can read an OD, can it read turbidity? So this ends up being, I didn't appreciate how complicated this was going to be, but the way the system is set up, again, picture is taken of a well as turbidity is happening, and then processing happens of those pictures to be able to identify turbidity and analyze those plates. Here you see sort of the difference between the signals that come out. So here you see me reading a broth microdilution plate, looking for turbidity in each one of the wells. And then here you see our research lab tech sort of looking at and identifying and reading the system that's there. So you can see it's not a one-to-one comparison, right? So it takes the computer eye to figure that out. And here is how it works. So light is shined through each well as it is growing or not growing bacteria. And then there are optical fibers on the bottom, same thing as the ELISA plate, to then pick up that information and transfer it through to identify growth of bacteria by turbidity or the blocking of light. So we built this into an incubator because the idea here is that maybe this should be more real time, right? If you're able to have an incubator with this setup in there, then you could have a plate with optical fibers driving from that plate, and then as that plate is growing, as turbidity is happening, it's able to read that turbidity real time. And a lot of machine learning had to go into it. So again, we're at the point that a signal is not just a signal. It can be very, very difficult, but we are at the point that the computing power that's necessary to read those particular signals is readily available, and we can transfer those signals to an MIC or a susceptibility report. So here's kind of what you would see in the grid lines. So here, growth standard, you have bacteria growing in the presence of an antibiotic, and it doesn't grow. So the antibiotic kills it. And so what you see over time is you don't see any blocking of the signal. But if the bacteria grows, meaning it's going to be resistant to that antibiotic, you're going to see growth happening. And so you can see these lines of growth because there's increasing turbidity in that particular space for that single well, for that single concentration of antibiotic. And so you can move this out over time for all of the antibiotics that have been tested to then create a antibiotic profile, a resistance profile, just based on what's going on in that plate and the reading. So what have we taken out? Well, we've made it cheap. So a lot of this is just in a simple incubator and is designed by plastics that are readily available and then laser printed. And we've made it automated from the perspective of not requiring clinical laboratory scientists or people who may not even be available in certain diagnostic areas to be able to read it. And when we compare for Klebsiella pneumoniae on 10 different drugs for 78 different patient isolates, we achieve an accuracy, sensitivity, and specificity that would clear as far as the FDA would be concerned for a device that would read susceptibility testing. And so again, it's this idea that we are working from a research and clinical lab perspective on developing new diagnostic, new ways of thinking about diagnostics so that they can become more available and we can reduce some of these access to care issues. And we have some partners now that are interested in this idea. So, and we have a graphic user interface, right? So again, this information is complicated. You want to be able to sort of seamlessly pass it to clinical providers so that they can have that information overall. So I think we started on the research side and sort of to finish up in the last 5 or 10 minutes. I want to tell you about what's happening on the implementation side from a lab equity perspective. So not just developing new assays or using new assays, but what can we do from a clinical lab standpoint to partner with healthcare providers to be able to use what we have already, stuff that's already FDA approved, but use it in a way that really expands access to care. And so I want to tell you about a program at UCLA Health called the Homeless Healthcare Collaborative. So Los Angeles, as many other cities across the country, are experiencing a crisis of homelessness, right? Because of housing crisis and overall inequities, we have a large number of people experiencing homelessness, and this number is growing. The challenge of homelessness is, of course, not having home and shelter, but it's also not having good access to health care. And so the way UCLA Health is approaching this is trying to provide health care to encampments and shelters across Los Angeles. And so I just want to sort of read some of this because it's very equity forward. It's a very equity understanding of the problem and how we should approach the problem. So at UCLA Health, our specially trained team members are committed to delivering care that is inclusive, culturally sensitive, and compassionate. It's a common misconception that drug abuse and mental illness are the primary cause of homelessness. More often, homelessness can lead to physical and mental illness. And the true causes of homelessness are unaffordable housing, inequitable access to care, escaping domestic violence, stagnant wages, systemic racism. All of these complex factors can come together to increase homelessness. So how do we provide care then at UCLA? So people experiencing homelessness we know are high risk, more likely to develop serious medical conditions. And so what we try and do is provide medical screening, whole family primary care, psychiatric care, housing and social referrals. But how do we do this? We do this using access vans. And so this is where lab testing is going to start to become critically important. So there are now 6 homeless healthcare vans that have healthcare providers in them that go out directly to shelters and encampments in the Los Angeles area. And so as director of point of care, this was presented to me as a project as how do we provide high quality diagnostic services in the setting of, let's say, a homeless encampment where the people who are there that are experiencing homelessness, you may not see again the next time you show up. So we don't have the luxury of taking in a sample, sending it to the laboratory, getting the lab result back. We really need to set up a direct point of care testing in a van, in 6 vans. This is a challenge, right? Some of the things that we had to think about, CLIA certificates, state certificates in California for being able to do the testing, which would clearly need to be wave testing. How do we train our medical doctors and nursing staff? What do competencies look like in the space around a van? Can we provide direct Care Connect access, right? For those working in point of care on this call, you know that just reporting the results can be difficult. They can end up in doctor's notes and in all these places. So how do we set up a way so that we can follow these results and track them if we want? What do QC logs and instrument maintenance look in a van? And I was especially concerned about this one, right? A van moves. And so you see this picture down here in the corner, sort of a point of care testing area in an urgent care that's just got all kinds of things all over it, right? You don't want machines falling down. Machines aren't made to be jostled. So what does your QC need to look like, and how often do you need to do it to make sure that that testing is compliant? And then additionally, how do we expand services in this particular space, right? I work in clinical microbiology, and there are lots of new molecular infectious disease point of care tests coming, including tests for sexually transmitted infections. How do we bring those on? How do we bring the infrastructure in to do molecular testing within a van? So again, all of these are very exciting things to challenge, but to me, all these need to be tackled by clinical laboratory professionals, right? We are the ones that need to put the plans together. We may not be the ones in the van, and that's fine, but we need the ones to put the protocol together to make sure that this is all very, very high quality. As healthcare reaches out, we need to reach out with that healthcare to provide excellent lab testing. So what are some lessons learned here? You know, I think that wherever you're working within the clinical laboratory professional infrastructure, there are lots of points of entry to lab access and lab equity. And I think you should go with what excites you the most, right? Clearly, I'm excited about this entire pathway. I'm less excited about the regulatory approval piece, but I'm very excited about novel assay component, clinical test development, clinical test implementation, and I try and partner with people that are excited about regulatory approval, right? Because it takes all of this. It takes this entire pipeline. If we're going to close some of that diagnostic gap that we talked about in the very beginning of this, and I'm excited that more and more people are starting to think about their work in this way, because you may have already been doing some of this work. You just didn't sort of think about it from a lab equity lens, and this idea that from the lab side, we can have a large impact on diagnostic equity and also overall on addressing healthcare disparities. So I want to finish there. I'd like to acknowledge all of these groups that I have partnered with or I'm working with directly in lab equity, and I hope you all have enjoyed this talk. Thank you very much.